Over the course of 50 years, Dr. David Gough has been a major contributor in the field of bioengineering and implantable glucose sensor technology. He has produced over 95 publications in the world's leading science journals, such as Science Translational Medicine, and holds 18 U.S. patents in the areas of bioinstrumentation and biomaterial. One of his most successful lifetime achievements is the implantable glucose biosensor for diabetic patients, which holds the world record for the longest stable sensor implant. David Gough received his PhD from the University of Utah in 1974, working on enzyme electrodes. His graduate studies sparked his lifelong quest in developing an implantable glucose biosensor.
When I was a graduate student, my advisor, Joe Andrade, showed me an article from Popular Mechanics that described the concept of the glucose sensor, and I got interested in making one. I used to work with William Gough on artificial organs, and so I actually built one and tried one for my thesis. As part of my thesis, I implanted 2 electrodes in a dog and measured their glucose and oxygen, but I didn't fully understand the sensor at that time.
After his graduation, Dr. Gough moved with his family to Boston, where he served as a postdoctoral fellow and adjunct professor at the Joslin Clinic of the Harvard Medical School.
I then moved from University of Utah to Joslin Clinic, and we actually built some sensors. We made a device for testing sensors, and we didn't succeed in using those in animals at that point either.
The biggest challenge with sensors based on enzyme reactions were the lack of enzyme stability.
Glucose oxidase is an enzyme that had been made since 1921. But no one had tested its stability at body temperature in solutions. We got some glucose oxidase and made it as a covering over the enzyme electrode and tested it, and we found that it was stable. It depends on the molecular conformation of the enzyme, of course. We then tested the enzyme as a membrane cross-linked with glutaraldehyde and found that it could be very stable. We then made sensors of all kinds with immobilized glucose oxidase.
The types of sensors that Dr. Gough built over the years varied in size, shape, and function.
I've got here a group of medical devices that I've developed over the years. This is a needle sensor that you insert in the abdomen, and it's got a small little region here for glucose oxidase, and it's an electrochemical wire inside, and you can plug this into a telemeter device, and you can record blood glucose continuously with that.
The needle sensors like this, like this, are commercially available. They're sold by Medtronic and Dexcom, and you implant them in the arm like this, and they have a small telemeter connected to them, which is in the form of a circle, and you tape that on the arm, and then they transmit signals for about 7 to 10 days.
Another type of biosensor Dr. Gough worked on was the lactate sensor.
After we had such great success with the glucose sensor, we decided to use lactate oxidase, which catalyzes a reaction between lactate acid and oxygen with immobilized glucose oxidase to make a immobilized membrane over lactate sensors. We use the lactate sensor in seals, where we go to Point Reyes in Northern California, a beach where a lot of seals congregate. We select a suitable subject, one that doesn't want to bite us, and we put the lactate sensors in the form of a small needle like this into the tail vein of the seal, and we connect a telemeter like this onto its outside, and then we send the seal off swimming They swim down past San Francisco, past San Diego, down to Baja, Mexico, where they have babies. And the whole time every time they come up for air, we can transmit a signal from a transmitter like this and measure their lactate levels. And some seals dive up to 450 feet. And that's how we made lactate sensors.
However, Dr. Gough's lifetime work was anchored in his vision to develop long-term biosensor implants that are functional for far longer than 10 days in the body. He continued testing the glucose biosensor at the University of California in San Diego in many animal models, such as hamsters, dogs, and pigs.
Then I came to San Diego, and we made lots of sensors of all kinds, this kind, all kinds of sensors. And we tested them in dogs. We published a number of papers with Joe Lucisano, Brian McKeon, Pius C., and Brad Sargent as co-authors. And we tested these sensors in animals and found that they could indeed last at least 3 months as implants and function effectively.
One of the first prototypes of the glucose biosensor was implanted in dogs for over a year.
This is an implant itself. This is an antenna which goes in the skin around here. One of these is an oxygen sensor, and the other is a glucose sensor. And these are implanted in the dog, and about right here.
As smaller batteries became available, the goal was to reduce the size of the biosensor implants as well. As the biosensor electrode discs were miniaturized to the size of a penny, microcirculation studies in the window chamber model in rodents determined the relationship between subcutaneous blood vessel distribution and the biosensor signal output.
This is a plate for a window chamber, which you use in hamsters. You couple both parts of this, screw these little screws in together, and then put glass plates on either side and pull up the skin on the back of the hamster and screw them in, and you can see through the tissue, and you can look at the microcirculation, and you can put a sensor in here, and you can understand the patterns of the microcirculation.
The understanding of how the biosensor reacted to oxygen fluctuation in tissues was one of the cornerstones for the development of the long-term implantable glucose biosensor, first tested in pigs.
We did studies in pigs where we implanted just the sensor without the enzyme membrane and measured oxygen.
Mimicking real patients with diabetes, the glucose biosensors were then tested in diabetic pigs.
The most exciting experience in development of the sensors was when we did our study in pigs, where we implanted the sensors in pigs that were non-diabetic for 6 months, then converted them with streptozotocin to diabetic pigs. Found that during the non-diabetic period, their response was completely flat, but during the diabetic period, that their glucose went all over. We published this paper in Science Translational Medicine.
Another challenge for long-term implants is the foreign body reaction that may induce implant encapsulation by surrounding tissues over time.
Most investigators who are familiar with implants thought that implant tissues would circumvent the sensor and prevent flux of oxygen and glucose to the sensor, make it impossible to measure glucose. Lucas Cumosa and our team implanted sensors in pigs and found that this was not the case.
With this success, Dr. Gough and his graduate student, Dr. Joe Lucisano, founded the company Glycens, with the goal of bringing the long-term glucose biosensor to clinical applications.
Then we founded a company in 1998, Joe Lucisano and I did, and We raised $13 million from SBIR grants, Small Business Innovation and Research grants. And we raised other monies from private investors, and we did clinical trials in humans, 5 patients, and found that we could measure the parameters of the sensor response.
They determined that they could make very accurate measurements with an algorithm they developed to analyze the signal.
Found that the response was very predictable. We developed several patents to measure the accuracy of the sensor, the longevity of the sensor.
With the first implants of the glucose biosensor in diabetic patients, Dr. Gough's lifetime achievement in long-term diabetes care became legacy. It is currently the only existing long-term implantable glucose measurement device that can replace finger pricking.
When we implant the sensor, initially the sensors like this, the needle sensors, only lasted for about 7 to 10 days. And Dexcom and Medtronic are still trying to make those last longer. When we implanted the sensors in humans, we first got 6 months survival. And now we get about a year. We expect to get about a year and a half in our upcoming clinical trials. When the sensor's finally developed, there won't be a need for finger stick companies because there'll be no need for finger sticking. It'll save a lot of money on federal expenses in health care. And of course it will benefit, most importantly of all, humans with diabetes.
After a long career as an outstanding engineering educator, Dr. David Gough achieved faculty emeritus status at UCSD in 2021. While his students will continue to carry his work to new frontiers, Dr. Gough's legacy will live forever among the fathers of bioengineering.